Yeah, my name is Hugo Magalhães. I'm a PhD student in Tobias Marshall's group in Germany. And as said, I'm here to present this work entitled Sequence to Graph Alignment-Based Copy Number Calling Using a Flow Network Formulation. So for roughly 2 decades, people have been assembling genomes into linear sequences and then comparing sequences against each other by aligning reads to those linear sequences and comparing them by doing so. But now in recent years, a lot of people, which I think includes most of us, have been exploring different ways of comparing and representing genomic sequences. I would dare to say that the most famous of all might be the graph representation, where we represent different sequences in a single structure, as we can see in this example, which will then allow us to better compare them against each other, and also removing the reference bias that we have in the upper part of the slide. In parallel, throughout the years, variation in copy number has been associated with multiple phenotypes. Here on the left side, you maybe can see that copy number variants in the RCL1 gene have been associated with a range of neuropsychiatric diseases. And on the right-hand side, you can see a deletion of this FCGR23 gene cluster that has been associated with lupus. So to detect these things, basically we need to call copy number. And copy number calling is defined by determining how many times a certain sequence appears. And in the graph context, this just corresponds to determining the number of node traversals. So, and what's a node traversal? Basically, since each sequence is spelled by taking a walk on the graph, the number of node traversals is just the number of times the number of times the sequence walk through each node. In this example here, you guys can see the corresponding copy number values that would be attributed to this graph. So copy number calling has been often done using normalized read depth from alignments against these linear references. So on the right side, you can see what would be the expected result, and we just do this by dividing the number of reads aligned to each sequence by the average coverage, and this gets us the copy number estimation. However, in this case, the linear sequence, the linear reference above only represents one form of the sequence. And if we instead use a graph for this, we can represent more sequences, and we get rid of this reference bias from before that I had mentioned already. So for example, in this case that I present here, since the reference walk through the graph includes, well, on the second bubble, node E instead of node F, the linear copy number calling will erroneously attribute a copy number of 4 to sequence C, where in fact, in this case, it's just that C and F are analogous. And so they have each copy number 2 instead of being node C with copy number 4. And this is something that we can address by using a graph instead of a linear reference. So if we just put the copy number values on the graph, this is what we get. However, if you look closely to this graph, and well, let's assume that we here have 2 sequences being aligned to it. If we try drawing the 2 walks of the 2 sequences through the graph, we can see that the first one, we draw it pretty easily. But when we try to draw the second one, there's no path that this yellow sequence can take to unite the left side with the right side. So this is because the copy number of node D is inconsistent with the rest of the graph. So what we are trying to do here, our goal here, is that by taking a pair of a graph in GFA format, together with the alignments to that same graph in GAF format, we want to call a copy number value for each node such that the read alignments to the sequence are used, as shown before, but also that the consistency throughout the graph must also be considered. So for that, we propose a solution consisting of these major steps. So we take a GFA graph and a GAF alignment file as input, as I mentioned. We retrieve node coverages from those alignments and then transform those coverages into probabilities. And finally, we feed those probabilities to a flow network in order to get our final copy number values. So first of all, we read and pre-process the graph to minimize the overlaps, such as we don't account for the same sequence twice. So let's take this example. If there's an overlapping sequence between node A and C, which corresponds to the edge between them, we will just get rid of that piece of sequence in one of them. And for this process, we use a set of hierarchical rules to decide on which side we will cut. Then from the reads aligned to the graph, we compute individual base pair coverages per node, such that the final node coverage corresponds to the sum of the coverages for each position within the node. We do this so that we can account each read for more than one node and also to overcome the variation in node sizes, as we are always considering size 1 for this case. So the next step is turning the coverages into probabilities, and we do that by assuming a negative binomial distribution of the coverage data, and we compute a probability value per node per copy number value. And this is done within a small range of copy number values that we define for each node. And just as a note here that we use the probabilities in natural log space. So our goal here is to maximize these probabilities across the whole graph. And we use integer linear programming for this. In this example I've been showing to you, this maximization will be very easy, right? We just take the higher probabilities, and that's it. However, what if we just switch these two probabilities right here? Well, then by taking the higher probabilities, we would bump again into the consistency issue in node D. So in order to address that, we formulate it as a flow network problem. We have a source and a sink, and we have flow moving through the graph from the source to the sink, making sure the copy number values are consistent. And to ensure that, we need some constraints. So the main constraints are the ones ensuring then the conservation of flow. So for every node, the flow entering on one side must be equal to the flow leaving on the opposite side. And also the sum of incoming flow and as a consequence of outgoing flow must be equal to the copy number value assigned to the node. However, we know that sequencing or assembly errors can still happen. Or maybe our sequence contains a new variant that is not present in the graph. So we can't enforce these constraints strictly. We have to allow the conservation of flow to be broken. So we achieve that by using this concept of super source and super sink. So super source and super sink have edges to every side of every node. And these so-called super edges can be used at a cost, allowing the conservation of flow to be broken. And just for the sake of the example, let's imagine that the probabilities really give a strong signal that node A has copy number 2, but nodes C and D have copy number 1. We could still do it by using this super edge from A to the super sink, sort of flushing away one unit of flow. Additionally, we apply two more constraints. So one of them is meant to penalize the use of the same edge in different orientations. This, in some specific cases, it might make biological sense, and that's why we penalize it but still allow it. But usually we don't want this. And the other one on the right side aims to favor the use of edges supported by more reads. So for example, in this case, the edge from C to D is not supported by any read, as you can see, so we would favor the use of the other 3 edges instead. So for now, we've been applying this tool for assembly graph quality control. For those of you that have never seen an assembly graph, this is how, well, a good one looks like. So most of these big components correspond to chromosomes, or at least most of the chromosomes. And I'm going to show you some results of the tool applied to a Virco assembly graph, so this graph. The assembly graph of this sample from the Human Genome Structural Variation Consortium. So for those who don't know it, Virco is a hybrid assembly pipeline developed for telomere-to-telomere assembly using accurate long reads, in this case, HiFi, together with ONT ultra-long reads, and this is what it produces. And so we then align the same HiFi reads used to construct the graph against the graph itself using Graph Aligner, and we then applied our method to this pair of graph plus alignments. So the tool, which is implemented in Python, ran on a single thread for about 2 hours and peaking at 3.5 gigabytes of memory. And this is basically the visual representation of the results, where the nodes are colored according to their copy number value. I hope it's distinguishable enough. And by looking at this, I mean, people can immediately get an intuitive idea of the results and what they mean. So we expect diploid chromosomes, which is the case for most of the components. But maybe to better explain it, I'm going to zoom in on 2 of the components so we can see what's happening more closely. So this is the first one. So this component actually corresponds to chromosome 11 of this individual. And as you can see, we have a diploid chromosome as expected, with these alleles in this side, the alleles on the bigger bubbles being haploid, and these common ones being diploid. But we can also see these red ones and these smaller bubbles. And what are these? So, sorry, these are actually misassemblies. We have manually checked these regions, and turns out that these 2 nodes in the bubble are actually the same piece of sequence, but the assembly constructor erroneously splitted them into 2 different nodes. And well, we checked with Virco's developer, and this has been observed multiple times and is a known issue that they are addressing. So as you can see, our tool was able to spot these places, these problematic regions, and flag them, even though all of these red nodes actually had coverage. So the second component I'm going to show is this one on the bottom. This is chromosome 2 this time. So once again, you can see mostly a diploid component with this putative misassemblies being flagged with copy number 0 on here, here, and all across this region. But then if you look at the left part of the component, you can notice that it actually becomes triploid. And the reason for that is basically the signals from these 2 these 2 regions where there's a accumulation of putative misassemblies that kind of mess with our flow. But still, we can still keep this localized, and we are still flagging where the problematic regions are and where people building these assemblies should curate their assemblies. So basically, we think that the tool has some virtues and one main challenge. So on the positive side, it produces consistent copy number values, it flags misassemblies, it's also able to genotype samples, and it's not specific to any sequence technology or graph building tool. But on the other hand, it highly depends on the graph topology, although we think about this more as a feature rather than a flaw. But yeah, it's still a challenge. So summing up, this work addresses the consistency issue by providing a new method for copy number calling on graphs. And as a consequence, it helps moving towards a broader usage of graph genomics. There's still a lot to do, so we want to expand this to more application cases, namely pangenomic genotyping, which is already in motion, and semantic copy number calling in cancer settings. And finally, there's a minor adjustment to be done in the statistical model that computes the probabilities, which we believe it will give us even better results. So I'd like to thank the Marshall Lab, especially Tobias and Timofey, which is talking after me, Guna and Jonas from Cloud Lab, and as well as all the institutions and consortia that supported this work. Thanks.
Questions? So is your method intrinsic to copy numbers only, or do you think you could tweak the formulation to maybe pick up misassembly inversions or other things?
Sorry, misassembly inversions, you said?
Yeah.
I mean, in this case, not really, because it doesn't. So it's blind on the orientation. So it's basically a matter of quantity of align reads, or in this case, align base pairs. So it's not for inversions. We would have to be aware of the actual orientation of the flow. But yeah, I can actually, the results outputted can actually tell you which path the flow is taking. So I consider this idea of maybe trying to output also the path, but for now that's not the idea. But I think it can be done.
Thanks. Great talk. I was wondering if, kind of similar to the question I was asked before, if it's specific to copy number, or do you think it would be possible to use this to convert splice junctions to isoforms using the same formulation of flow in RNA-seq?
I mean, to be honest, the tool gives you the number of copies of the node, but the interpretation you do of that is not necessarily so copy number variation setting, so a genomic variant or allele, so different alleles. The interpretation, I guess, it's on the user. That's why, for example, right now we're using for assembly QC, where we just are trying to see where actually there are assembly errors, and we were not looking for actual copy number variation or something like that. So in that sense, I think it's up to the user's interpretation.
I really liked your talk. One question: how dependent is this in terms of distinguishing the original nodes A, B, C, and D? So in other words, these loci tend to undergo conversion back and forth. Sequence identity gets extremely high, even though the locations can be very different in the genome. Does that method require that A, B, C, and D be distinguishable?
I mean, isn't that more dependent on the alignment itself?
Sure. I mean, that's the nature of like FCGR3 or complement factor 4, right? The copies are so identical that they approach allelic variation. So the question is, it's not just going around the graph multiple times. It's being able to distinguish which copies, because some are pseudogenes and some are genes. But when you get to sequences that are so identical, can it work if you have high identity, which is the most common state for normal polymorphism?
Yeah, so I get, you mean like if a piece of sequence is splitted in, or the repeats are splitted into different nodes, if my tool would be aware of that and would be aware if it's the same sequence actually? No, I mean, that's why I said that the main challenge is that it's dependent on the graph topology. So here, I think my excuse would be to blame it on the graph construction. But yeah, it is a challenge, and we have been thinking of maybe in the future, not for the immediate results that we are achieving right now, but maybe trying to do something that would change the graph topology and maybe work on that as well. But yeah, in this case, I don't even look at the sequences. I look at the alignments, and it's position based of the alignment. So it's blind on the sequence.